Part A, Basic Setup. Shown are all the equipment and chemicals you will need for this experiment. Cut the chromatography paper in half, like shown on the right. Then cut each of those pieces into half as well. Mark on 3 of these squares with a pencil a line approximately 1 centimeter above the bottom. Part B, Performing the Experiment. In order to determine what substances make up these 3 mixtures, we're going to use 7 FDA food dyes that we use to make these mixtures. These food dyes are red 40 through green 3 and are shown here in these vials. We will also use these in order that is shown here. Place a sample of each one of the FDA food dyes and mixtures onto one of your 3 chromatography papers by using a piece of paper cut to a point, dipping that in the solution, and then placing a small sample interdispersed throughout the line at the bottom of the chromatography paper. Continue this process for each sample. Here are the three mobile phase solvents that we will use in this experiment. Water is polar, acetone nonpolar, and ethanol somewhere in between. We will use differing solvent polarity in order to provide different separation patterns of our substances on the stationary phase of the chromatography paper. Choose a solvent and place a small amount into a glass beaker, making sure that the volume height will not be higher than the pencil line on the chromatography paper. Allow the solvent to absorb up the chromatography paper. Once the solvent front is 1 to 2 centimeters from the top, remove the chromatography paper from the beaker to stop the movement of the solvent front any further. Part C, Data Collection. The samples I'm about to show you are from a previous run that was done of the same experiment. All of the same samples are present, they're just patterned slightly differently. To process the paper chromatography experiment, first you want to measure the distance the solvent actually absorbed up the chromatography paper. So measure from the top of the absorbed line of the chromatography paper to the original pencil line and write that down. Next, mark a line for the distance traveled for each substance on the chromatography paper. On the left-hand sheet of paper are each of the 7 FDA food dyes, and on the right-hand paper are the mixtures 1, 2, and 3. For these samples, you want to mark a line for each different color that you see present in the sample. Using a metric ruler, record these distances and write them down on a sheet of paper. Here are the results shown for using the solvent water, the results shown using the solvent ethanol, and the results shown using the solvent acetone. Part D, Data Analysis. Using the distance of the solvents traveled, the distance of each substance, calculate the retention factor, or RF, for each substance. You can use this value to compare the mixtures with the known substances for identification purposes. You will write a formal lab report on this experiment. Contact your group that has been previously assigned to you and determine what each member's contributions will be to this lab report. Download the attached Excel file with the RF data and images, as you will need both of these to input into your formal lab report. Once your group has finished writing your entire formal lab report, use the instructions to submit it on Blackboard.